Opening, well, nothing specific. I think we could maybe get into talking about some of the things that were brought up, and I would like to sort of go back to the survey results, the IQ survey results that were presented, which by the last speaker, which indicated that companies are using these tools, but they're not submitting them to the FDA, which is something that we are also observing. They're not submitting them as much or that much. But I would like to sort of thank the speaker from Hesperos for showing us that there are no physical barriers to submitting these types of studies to FDA, because clearly if they're not being submitted, it's not because FDA is preventing people from submitting them, but it's because people are not voluntarily submitting these studies to FDA. And so I think that what maybe I would like to start focusing on is really what are some of the things that people think FDA could provide to sponsors that would encourage them to submit the data that they have, because clearly this type of data is being generated in the discovery stage for both safety assessment as well as for efficacy. Some of the efficacy examples were presented today. And so I was kind of hoping that maybe people would address the specific, maybe, 2 or 3 types of things that you would like to see in the FDA guidance document or something that would encourage you to submit more complex in vitro method data to FDA, basically.
Great question. So perhaps, Deidre, coming from previously working at GSK and your work you've done there, any perspective that you'd like to add right now? We can go on to the other.
Okay, so there you go. Based off of the various different studies that we've done and what I've seen in the past, I think that, I'm trying to understand what the FDA will, generally do with the data, and really having that, confidence that when the data is submitted, that it is not going to, I would say, per se, have a negative impact or on, what you're doing. I think earlier today, there was some mention of the fact that, you would have the animal models and you would have the complex in vitro, model type data. And in those instances, knowing that these, just say from a safety perspective, knowing that these models are getting developed, when that data, obviously, when you generate the data, you're going to submit the data. And in some instances, it might not correlate, I would say, with in vivo data that you per se see from the animals. And then, understanding, how that would be treated, and would, that in vitro data really throw a hurdle in front of you, even though, say, your animal data might be showing something that is, I would say, positive in the sense of being able to move forward, but maybe the complex, data might not show that. So would that cause, a concern and cause a delay in the progression? So I think it's just trying to understand the implications of that and how it would be treated.
I mean, I think that right now the problem is that we don't have enough experience with seeing different types of MPS data or any complex in vitro method data submitted to us. And I think that what we've observed is that in those cases where there are some data submitted, you're right that the animal data is also submitted, but the CIVM data is not there's no justification or context of use provided. It's just included in the submission without really saying why it is that it was submitted, and also the amount of I'm using the word validation. I don't mean it's formal validation, but the amount of characterization of the method is not as robust as what you would expect in terms of trying to define some of these characteristics that were mentioned, or criteria that people would look at, cell source media, other types of things that are considered typically standardized. So if you submit your data and you have a justification for why you're submitting it, for what purpose it is supposed to be, used, and how it's supposed to be interpreted, and how you are demonstrating your confidence in the data, then I think those are going to be things that we're going to look at. And so, I mean, I can give you one example that from our own experience, we know that with one of these complex in vitro methods, if you're looking at ALT levels, for example, the enzyme activity and the protein levels don't always correlate. And so the question is, if you've measured that and you are reporting enzyme activity, but actually if it's not stable, how am I to know that you measured the right thing there? And so those kinds of things can be very specific to a platform, and you need to do a certain amount of work to demonstrate that there is a certain amount of rigor to that data. And I think if that's demonstrated, I don't think that there would be any negative impact, because it's purely going to be viewed as additional data that's added to the existing package of information that you're providing.
Can I just chip in there a little bit? So I think one of the sort of trends that we see in a large CRO like Charles River is the sort of increased activity of ultra rare and N=1 projects. And I think because many of those programs are running through professional CRO organizations like ourselves, you end up with a lot of very well-developed, complex in vitro model systems that aim to replicate the efficacy effect, mostly using azos. And I think you're going to start seeing a lot more of that type of data submitted with in IND packages. And I think that will come with the professionalism and the sort of clear context of use and the clear validation methodologies behind it. And hopefully that's one of the things that will encourage the industry to sort of move in that direction, because the individuals, the patient foundations, the small biotechs submitting that are just not going to be as understandably risk averse as large pharma. They have a very, very real reason for wanting to get that into the clinic and as quickly as possible. So I'm just sort of suggesting that I think some of the barriers could come down over time, expressly because of those types of projects.
I certainly agree with that. And I think one of the things that we're trying to work towards is to encourage people to submit things to us. So we also develop that level of confidence and that they can submit more things to us. And I think the goal is not to be punitive and to sort of say, oh, this data doesn't match with this other data, and so this is going to be bad for you. But it's more for us to have some level of familiarity with the type of data, since it's going to be, these models are very complex and likely to be very different depending on who is submitting them and how they did their assays and the sources of their materials and everything. And so it really is important for us to kind of get both data sets, the animal and the sort of CIVM, together for a period of time until we get a certain amount of comfort with that data. And I think that that's where we are going to be able to form some generalizable concepts that we can all agree on, which is then going to shift the balance towards depending less on maybe animals and more on in vitro in certain situations.
I see. We're already developing those complex in vitro models. We're working with many different worldwide companies, and they have confidence in them because they're submitting our data without animal data. So we're already there. It's not like in the future, it's happening now. And we're not the only company that is doing this. There's companies besides Hesperos. But the important point is, especially for the small biotechs and even the virtual companies, is we're doing it at a much shorter time frame and a much lower cost than the animal models would be. So it really fits into their models because, a lot of these systems that people might work on, if you have very, very large teams and everything, is they become very expensive. So what we're trying to do is keep the cost down and keep the time down. Okay. And that really is working well with the biotech partners that we have.
Thanks. Any other comments on that question, or should we move to a question in the audience? I think Mike has one.
Yeah, I mean, I think this is relevant from thinking about this with qualification as sort of the end game, right? If we're trying to come up with a model system that you can use and application across a number of different programs, I think Nakissa's point is important because to get that experience, we do need to see those data. And, I understand the concern and the regulatory risk that's perceived from submitting those data to FDA. But perhaps there might be programs that have been shelved or other situations that we could potentially use to learn from, where we had a positive animal tox signal, as well as some other data that were corroborating that finding, that could at least begin paving the way toward something that would look like qualification for some of these model systems.
Just two things on that, which is one, we are funded by the NIH right now to develop the NMJ model as a Botox tool, okay, to replace animals. And that's funded. Dimitri's my program manager right over there for that. And so the goal is to get an MPS NMJ model as a DDT tool for Botox, okay? And so that's already ongoing. We hope to do that pretty soon. But the other thing is that for rare diseases is that in many cases there are no animal models. Okay. And so this is the only system. And that's why we want to have that clinical correlation, because we believe we're getting acceptance because the FDA can say, oh, conduction velocity, I can measure that here. I can also measure that in a person. And I think that's the confidence that we're gaining with these systems so far.
And in areas where we have very active drug development, it could be very helpful to have those data from, a couple of different programs to be able to use them from one to another, and it has a broader benefit. I think one thing that may be a difficulty and sort of part of the reason for establishing the voluntary genomic data submission many years ago was out of concern for this. There was a concern that FDA was going to overreact to anything that was submitted. And I think that ended up not being the case. We're naturally very skeptical of new things, so that ended up not being the case. But with continued experience and engagement with FDA on these things for individual programs, for bigger issues like safety and whatnot, the questions, and we did this in a firewalled way, such that the team that was looking at the voluntary submission was not part of the regulatory review team. So there was a little bit of protection there from that. It was insulated. But what ended up happening is the programs ultimately started asking questions that should have been coming into the IND formally with review by the entire team. And it just sort of naturally progressed that way because everyone started getting comfortable with the technology. So, I think there is something to be said for potentially either a safe harbor or some other voluntary mechanism potentially.
And Mike, do you mind if I ask one follow-on question to the voluntary submission with the pharmacogenomics about how much data did you look at on the firewalled side as you were getting an understanding and confidence?
There was probably, it was not a voluminous program. It's there was probably 60 or so meetings in the first several years. So we had a large number of submissions early on, and then it began tapering off, and it was being channeled either to critical path innovation meetings or to the IND formal meetings. Individual data sets coming in from companies, they were all asking different things. So it's not really like there was something specific that was in this context here we're talking about today. Potentially, like there wasn't a question of qualification. It was how we're generating these data, how is it being used, what are you guys going to say about it, and those kinds of things was sort of what we got out of those meetings mostly.
Thanks. I think we've got one question in the audience right now.
Andre had a quick comment to add on to that a while ago.
Yeah, sorry, just to point out for the first question is that regarding that the data is not being submitted for regulatory submission, the IQMPS survey was published in 2024, but the questions that they answer were done way before. So things might have changed when you hear this this comment.
Thanks, Andre.
Yeah, I just want to emphasize the last points that were raised by Jim and Mike. The future is now. It is happening. And the notion that somehow you can't use these methods if they're not qualified is not true. The value of having a public-private partnership conversation about the methods is that, sure, it can provide an avenue for a formal discussion through qualification submissions, and that provides a lot of in-depth evidence discussions, but the discussion alone allows the better understanding and alignment of consistent messaging from FDA to industry and from industry to the FDA about the unmet needs and what they are seeing in their pipelines. countless examples in 30 plus therapeutic areas where we've seen that happen over 20 years with biomarkers, with clinical outcome assessment tools, with computational models, with clinical trial simulators, etc. I don't see why that cannot happen with CIVMs and NAMs in general. That the future is now, and we have this unique opportunity to leverage the public-private partnership model to take a leap and advance the adoption of CIVMs and NAMs. So just wanted to emphasize that important point.
I just one point to follow on regarding the safe harbor concept. I mean, to date, we've had very positive experiences of having early conversations with the FDA around new concepts. We've done that in sort of in silico methods. We've done that in complex in vitro methods, just having high level conceptual conversations that sort of feel pretty safe from our perspective so that we can plan for the future.
I just wanted to make a comment about the sort of qualification point that was just brought up. I wanted to clarify that there's absolutely no requirement for any complex in vitro method to be qualified before it's submitted to the FDA. Qualification is absolutely not necessary. It's a mechanism that exists if somebody wants to qualify their method so that other people may then use it for a specific context of use. But for a particular submission, for the purposes that a sponsor wishes to use, CIVM, they can use whatever method they want as long as they justify why they used it and provide some level of validation about the about the performance of the assay to ensure that you can trust the data. So I just wanted to kind of highlight that.
Can I just pick up right there? Is there value added though for the IND review if a complex in vitro model is qualified? Is that going to change the speed or the level of acceptance, or the readiness of acceptance of that data from your perspective? And I have a similar question for Jay. The qualification, is it obviously necessary in order to have interest from outside companies? But I wonder, is there value, or could you speak to the value that they might perceive if you actually had these qualified? So I'm missing the value of this whole qualification process. That the examples we heard were, is it necessary? Okay, that message is sent, but what is the value added? I don't quite get that.
The reason why, I assume you said Jay, right? When you, yeah, okay. Make sure I was answering in turn. The nice thing, especially for the Botox program, is that these people, the way they create the botulinum toxin is in a batch method, so they really can't quantify it except in an animal, or there's also some enzymatic tests they might be able to use that aren't as good. And so doing a lot of these things at once, okay? There's many different companies doing large batches. So to have to go in and provide the validation data for each one of those would be a chore. So if we can actually get it, submit the validation data, get it qualified, they won't have to do that, okay? They'll just be say, we're using the Hesperos method. It's already been reviewed by FDA. If we follow that process for the evaluation of theirs, they don't have to do anything else. So it'll lessen their time, lessen their cost by having a qualified method in the system, right?
I agree, but that's for a very specific context of use. So, and it's for something that's repeatedly going to be done the same way. But for, let's say, the LOI that York mentioned this morning, that the one that FDA recently accepted into the program, it's a very narrow context of use. Something like that, obviously, is not going to be very useful to everybody. And so for something like that, you may want to ask yourself whether you want to qualify it or not, because if you're only going to be using it once, there's no qualification necessary, because you just, say that your assay is capable of measuring what it is that you want to measure, that the data are dependable, and you don't need to qualify it. So I don't think that qualification is going to add anything in that particular perspective. But if it's for something like, Botox, or if it's for like, endotoxin or something like that, where you're going to check it over and over, then clearly having a qualified method is potentially useful. It might save time and money. And also, I think that any one piece of data is not going to be looked at by itself. It's going to be looked in the totality of the evidence. So I don't think that any study alone is going to say like completely, kill a program or save it altogether. So
I'll add briefly if I can, and I'm a bit out of my swim lane here because I don't review non-clinical toxicology data or anything like that. But the whole purpose of qualification is consistent interpretation across all therapeutic areas, right? So when and if companies are doing things in different ways, you're tweaking the parameters a little bit, then that starts inviting questions and you undermines the reliability, and that ends up kind of adding time and complexity to some of the interactions and how we go about interpreting the data. So to the extent that qualification provides kind of a baseline set of parameters that are consistently applied across programs, that eases that interpretation threshold from our standpoint. So there is value in that.
Yeah, thanks, Mike. I think it's a really important point to bring up around qualification, is that it's meant to provide confidence in the qualified context of use, as well as we gain more understanding to potentially apply it in different manners as well. But it sets a baseline for how it's applied. I think we've got a question from our virtual audience.
Yep. Question from the virtual audience. Would FDA create a guidance document specific to CIVMs in order to demonstrate FDA interest and commitment to considering or advancing them, and also publicly share cases in which CIVMs have been accepted, such as via a web page?
Yes and yes. We're going to do both of those things. So yes, we have definitely an interest, and we're motivated to do both things. And so we're currently working on both.
You guys are keeping us busy, basically what Nakissa is saying. But I want to kind of take a step back. I think I'm hearing lots of talk, but I think I want to let everyone know that, I think we're going to touch on this tomorrow, but there's a difference between using, I think, NAM, CIVM for efficacy and for safety, and it's not the same. And, for the most part, efficacy I'm talking about human pharmaceuticals is ultimately determined in people, so these early kind of in vitro efficacy or animal models are helpful and useful to determine, probably more so pre-FDA, internally at a sponsor, at a company, if it's worthwhile to take that compound to the FDA to submit an IND to see if it's going to go forward. But from the FDA side, once we get that application, we're, heavy on the safety, because, we need to make sure that this we're talking first in human clinical trial, never been given to a human being before. It's safety, so we need to, the efficacy is important, and it's important along the way, but it's the safety is what is important early on. So I just want to, keep that in mind, that, efficacy is ultimately determined in clinical studies, not in vitro or in animals, except for those rare instances where we're talking about like the animal rule, and then we're talking about, compounds for anthrax and smallpox and things like that. But for everything else, it's ultimately determined in clinical trials.
Yeah, Amy, thank you for bringing that up, because I think that, what Amy's saying as well is that for the safety studies, people are hoping to replace the safety studies with that. With the efficacy studies, I don't know that anybody's going to be replacing a clinical trial with that. So I do think that, there is a bit of a difference. We're being asked to kind of like, consider a set of studies for which we have very little information and sort of confidence in replacing, years and years of information. Whereas for the efficacy stuff, while it's useful for like rare diseases and for confirmatory evidence, they're not likely to replace human studies. And so it's going to be viewed very differently.
Yeah, I think also on the efficacy side, I mean, it's something that will help refine studies in the future, which may lead to either smaller initial trials, which would benefit in terms of time and cost, for development is another aspect. I think that we haven't really touched on so far with the efficacy component, but definitely on the safety side, it's setting a different bar. I think we've been waiting. I mean, one moment, Jay, for I think Andre had a comment as well. Is it still applicable, Andre?
No. I already made a comment before.
Okay.
Well, see, one of the things a lot of customers are working with us on is stratification of their clinical trials early, okay, and then utilizing that data in their clinical trial application, okay, and that's working out very well because we can see those differences in human-based models, okay, that are having direct application for what they're seeing in their clinical trials, okay, and so that's a big difference in what we're seeing because again, I don't know who gave the example of oncology. It was you, I think, and just showed that the first back in 1998, when you had a biomarker for cancer, you're able to limit down, the population to the point now where all cancer is done that way, right? Hopefully, we can get that with just about every disease. We're trying to do it for Alzheimer's. We've already doing it with ALS. We're trying to do that with a large number of diseases. So if we have the clinically relevant human models beforehand, we can start adding that data or limiting what goes into the initial IND to be able to increase the success rate of that clinical trial.
And I think I would just add to that point, which I agree with. I think the importance of sticking to truly functional readouts is where that's going to be most effective, which is primarily what Hesperus are doing. And a number of other groups in this space are focused on functional readouts, not just biomarkers.
Thanks. And it looks like we've got a question from our virtual attendants.
Yep, this one's from Monica, who presented earlier. What is the state of play of CIVM in CROs? Is there interest in including CIVM in their pipelines? Are your clients interested in CIVM services?
Yes.
And Monica, we're a CRO actually.
Yeah, there's no question. And I think it's sort of one of the things that we've been strategizing around for the last year is how we play in the ecosystem and how we help accelerate the use of CIVMs in both efficacy and safety. So I think being a large player in the space, we actually have some sort of influence over level of adoption. And we're also prepared to be a qualification partner for those innovators who need that.
I'll bring up one, additional question. I think that, we're talking about, some functional readouts that are, known in humans that you can do in those in vitro models. I would say there's also those questions where there are no biomarkers or there are no really good translational models for, say, a specific type of safety. So when it comes to really generating these, in vitro models and trying to make them most human relevant, if we don't have, say, a biomarker right now to follow, and, just say from the pharmaceutical development side, we're stopping development of compounds because we don't specifically have that, then generating, I would say, these sort of, context of use using a model that's, say, for vascular injury, I would say that is showing that it can recapitulate the vascular system in human. But how do you then go about being able to say that you can use such a model to be able to, move your compounds forward when there are really no known, say, animal biomarkers or human biomarkers for that? So I think the space has, some room to grow in those instances where you don't have that functional readout that's really helping you develop and show something that's safe. So perhaps, these models are actually more relevant, in incorporating the, right cell types into these human models. So it's just, a big hurdle, I think, in those instances to overcome.
We could keep going if you like.
Well, I was going to see if you had additional questions you wanted to ask each other right now as well.
Well, I was just going to add one other thing in addition to what Deidre said. I mean, I think the, another thing that we often wonder about is the exposure relatedness of a lot of safety issues. I think that could also probably be potentially used. Right now we often rely on concentration response modeling out of the clinical trials, which there's often few cases in clinical trials if the program is successful. So there's always a dilemma, trying to understand whether it's modifiable through dose adjustment and those kinds of things. But I think there are definitely pockets of information that could be very helpful.
Yeah, and I just want to make a sort of a comment, which might draw some questions, but I don't think we're ever going to get rid of animals entirely in safety because we can't reproduce systemic tox, right? It's just never going to happen that I can tell. I'm pretty imaginative. I can't see that ever happening. And there's other aspects of safety I think is going to be very difficult with MPS. So I'm not advocating total elimination of animals in these systems. But I do think there are ways you can complement to reduce, and there are systems, especially with efficacy, where you can replace. But I want to make really, really clear, we're not advocating for entire removal of animals in the process. I just don't think that's possible.
Yeah, speaking on behalf of Charles River, I would wholeheartedly concur with that. We don't believe that you can one day have a suite of complex in vitro models that will be a surrogate for whole organism tox. But we do believe that you can develop CIVMs around maybe very specific toxicities that you can model effectively and add into an integrated system and maybe just better predict human toxicity at the end of the day. And so those are the sorts of assays that Charles River will be focused on developing with the innovator companies, for instance, around gene tox.
Yeah, I think that's a very good point, because that's when we say that the model has to be as good or better, and really preferably it would be good if it was better in sort of providing additional information that we currently cannot get, and to better predict potential toxicities in the future.
Thanks. And I think, Julie, along those lines, are you looking at integrated services right now that combine both in vivo and in vitro to assess pieces when you have common issues that aren't easily answered?
Yes. So, I mean, a lot of people don't think of Charles River as a discovery organization, but we are, as well as a safety organization. So we cover the gambit. So all of the conversations around using complex in vitro models for efficacy, early reads on efficacy, and then supporting efficacy into IND packages is relevant to what we do. And then, yes, we are somewhat limited in what I'd call our in vitro tox today, but we recognize that the true future of decision-making, say 10, 15 years from now, is likely to look different and requires us to have a different set of capabilities, not just complex in vitro, but also in silico and predictive toxicology. So that's sort of future gazing for our organization. It's one of the reasons I'm here today.
So that just made me think of something, when you asked that about the integration, do we think that maybe we can think about qualification as not just an in vitro method to replace, but we submit it with animal data to say this is how it can complement to meet the other two R's, the reduce and refine. And so maybe that might be an easier thing to address, and comparing it to how we do it traditionally with more animals, do it with less animals, but now adding something in addition. And so that potentially I can see that there might be some value in trying to qualify an integrated approach where you use less animals, but incorporate one or more CIVMs into the whole process.
I mean, one thing I do want to point out is we worked with L'Oreal for many years to develop, and they were not allowed to use animals after the 2012 in Europe. And what they did, they worked with us, and they do have a complicated suite of systems now. And what they use is in silico, then combine them all together, then predict the tox to go forward. And so, now obviously the cosmetics is not as high a bar for safety. But there are models that combine the in silico and the complex models, some of which we developed for them, that they're utilizing right now, they feel very confident in. So those kinds of things are possible. Okay. It just has to be, how creative can you get?
Yeah, and I think, with the launch of the complementary program, I think it's going to be interesting to see how those groups also begin to further integrate across different NAMs and apply those in the future.
Hi guys, Danny from Emulate. I want to go back to the beginning discussion, talk about barriers and pain points. One of the things that's clear about where we are now after having 10 minutes of discussion is that safety and efficacy are very different in this regard, right? With efficacy, it's pretty clear that sponsors have the motivation to submit ad hoc data to help the submission because it's to their benefit to push this forward. With safety, we're seeing the opposite, right? With safety, we're seeing a hesitance for people to submit data because why confuse the situation? Your animals were clean. You don't need to add more data, right? Why bother? But the push here may not need to come from sponsors. The pull rather might need to come from the FDA because the FDA has the remit to protect people. The pharma company, the clinical trials are going to catch the toxicity, but FDA needs to make sure people don't get poisoned in the clinical trials. So with this split between safety and efficacy, again, efficacy for sure, ad hoc submissions, let's wait for the industry to catch up. They'll do whatever they need. But with safety, shouldn't the FDA be proactively pulling this technology in because of this remit to protect people?
You're saying that these kinds of models are likely to protect people by, I agree with you. If you can demonstrate that they can, and it's, you can justify and provide the data to support that, I certainly agree with you.
Right. So several companies have done that, right? Whether it's spheroids or organs and chips, liver specifically is where it's been the tip of the spear. And people have shown that we can catch toxicity that animals miss. Nobody's saying replace the animals. You're just saying add another level and a belt and suspenders, right? Another check in the system. You're not letting in anything that you wouldn't let in before because the animals will still catch it, but you have another layer of protection, right? Again, multiple companies have done it for liver. Isn't this a call for the FDA to say, here's an opportunity to protect people?
I mean, you're right that the animal studies have not been very good at protecting, but the drugs that have been recalled for idiosyncratic, the humans were not good at predicting it either, so otherwise we would have predicted it. Yeah. So I don't know that doing these additional studies is actually going to catch the type of toxicity that you're looking for. So I think that it depends really on what your sort of endpoint is. I think if it's an endpoint that clearly, even humans would not be able to, catch it, then certainly, yes, you should try to do it in vitro if possible.
Yeah, and I agree. I mean, if something isn't caught by in vitro, don't bother using in vitro. If it's idiosyncratic and rare, you're never going to catch it. Don't bother trying. But let's focus on Sphero, just so the focus isn't on us. They showed that it can catch a lot of drugs in a regular panel that were missed by animals that are not idiosyncratic, that made it in the clinic and ended up poisoning people. Right? There is a body of evidence that shows that there is an opportunity to be more protective.
Right. But I also think that there are limitations in terms of the length of the study. So obviously, the animal studies, you can do them for several months, and that is just not possible right now with the in vitro. So I think part of the issue is to try and predict some of those more longer term toxicities. And so there are challenges. I'm not saying that it's not going to happen. I think that definitely these are things that we need to think about to try and specifically incorporate into.
Right, but to go back to my initial point, there are questions, but I'm arguing that as opposed to efficacy, where the push is in pharma to try to get, the motivations on them, I'm arguing that for safety, the motivation should be on the FDA side to encourage this. Whereas what's happening right now is actually a reluctance to submit this data because of the fear of unexplainable data.
Right. So, yeah, you're absolutely right. And so that's what we're trying to do in response to the question about the guidance document. We're drafting a guidance document specifically to try and encourage people to submit more data to us, because by looking at the existing data, it's very sparse. And so it's very hard to draw trends and identify sort of best practices from that. We need that type of information in order to be able to inform sponsors. And so that is what we're hoping to do. And, it's going to take time, obviously, unfortunately.
So we were told the FDA would not consider a safe harbor. Can you mention one or two ways in which you can coax data from people who are reluctant?
I think that basically by people submitting data in their submissions, in addition to the animal data, we would hopefully want to review and compare ourselves and see how those data look. And over time, we would get the sort of confidence that's necessary, unless other people want to do that.
Right, right. And again, the challenge with safety is that they don't want to run these studies in the first place. It's not just a matter of not submitting them because they're afraid of the consequences of additional data.
I mean, they are running. I mean, they're all doing the animal studies anyway.
You know, we're coming up at the end of this portion. There's one brief response and then
Well,
one last question from Mike.
I think Danny you just identified a barrier, because you said that, well, you can't run these long enough as long as the animals. You can. Okay. And so we as a community need to show that we can do that, because we can get them out to months. Okay. And so so you need to sort of say what you think they can't do and put that out as a challenge for us and see what we can do. But you just identified at least 2 or 3 barriers in your reply to Danny that said, well, no, we can do that. Okay. In certain instances, we'd like to know what you think they can and cannot do. Okay. And give us the challenge of overcoming that. That would be wonderful.
Thanks, Jay. And one
Just
one last question.
Ivan Rusin, Texas A&M, just quickly to follow up on this point. I think we're all comfortable with positive responses in animals and in vitro models. And I think what sponsors are really looking for is, can anyone trust the negative data, or should we keep testing until we actually get a positive result? So I think it cuts both ways, and I'm really glad that our colleagues in the complex in vitro models are actually advocating for animal studies. Both Hesperus and Emulate seems to, advocate for that. So I think FDA is trying to protect us, and then sponsors will test as much as they feel necessary, but we're probably all not ready to make decisions on negative data just from complex in vitro models.
Yeah, good. Thanks. Good point. All right. Well, thank you everyone for participating in